to ensure that these two chromatids are held together. They don't go, wandering in the nucleus until the time when they come to separate. It is also the region that you're going to see where the protein fibers that pull these chromatids apart also start forming. So most of its role is structural rather than coding for proteins to make, functional products in the cell. So among the features you're going to in a chromosome are the telomere as well as the centromere. These normally are just for structural purposes, protection and so on and so forth. Now, talking about the centromere, it is not always at the center. The centromere position can shift. Sometimes it can be close to this end, sometimes it can be close to that end, and sometimes, yes, it can be at the middle. So the chromosome itself can be divided, okay, into what is referred to as a P arm, which is like a short arm, and the Q arm, which is like the long arm. I know this doesn't make sense because this looks like this end is long and this end is short, but this is just what it means. P is for the short arm and then Q is for the long arm. Now, what is the reference point here? We're trying to refer to where the centromere is. So based on where the centromere is, that is to say the distance left on the P arm or the Q arm, you can divide the centromere into about 4 different chromosome types. So sometimes the chromosome can be referred to as subcentric. In subcentric, the centromere is slightly off the center, creating some longer arm and a shorter arm. In subcentric, you are referring to the position of the centromere in regards to the P arm and the Q arm. You can see that in subcentric, it is somewhere off the center of the chromosome. So this chromosome is referred to as a submetacentric chromosome. It can also be referred to as metacentric. Metacentric, this is when the centromere is located midway of the P arm and the Q arm. So what are we saying here? The centromere position can vary on the chromosome. Okay. Now, when the centromere is off the center of the two chromosomes, you say it is submetacentric. Sometimes the centromere is kind of at an equitable distance, giving rise to an equal P arm and a Q arm. This kind of a chromosome is referred to as metacentric. We also have telocentric. So in telocentric, this is where the centromere is at the very end of the chromosome, where you almost have no P arm. This is telocentric. It can also be referred to as acrocentric, where you just have a very short, P arm. Not really at the end compared to telocentric, but it leaves a very short end of the P arm. So depending on where the chromosome is placed, these chromosomes can be referred to as telocentric, metacentric, subcentric, submetacentric, and so on and so forth. And this is important for us to know because it helps scientists who study chromosomes understand even what chromosome they're looking at. We're going to see in the next few slides why knowing this is very important. So what are we moving on to? We have learned that chromosomes occur in pairs. That is, in us, yourself and me, we're diploid organisms, and that each chromosome has characteristic features such as the centromeres, the telomeres, and so on and so forth. But how do scientists study all the chromosomes in a cell at the same time? Well, they do that by taking a snapshot, like a picture. Most of us today, by the end of today, I don't know how many pictures of ourselves we're going to take, like selfies. Well, chromosomes can also be studied or understood in the same way by taking a picture, like a snapshot of all the chromosomes that are found in a cell. Let's look at this picture of chromosomes that is referred to as a karyotype. It is the complete set of all the chromosomes in a cell. So this looks like a chromosome. This looks like a chromosome of a human being. When you look at the different chromosomes, so this is a position where the centromere should be. If you inspect all these chromosomes, you're going to see that some of these centromere positions could actually be different. Sometimes the Q arm is longer and things like that. This is what we actually saw in the previous slides. So by looking at the centromere position, scientists are able to tell, for example, which one is chromosome 1, which one is chromosome 2, and just be able to also arrange them, because we understand one of these chromosomes is coming from the father, one of these chromosomes is coming from the mother. This is chromosome 1. When you go all this way, we know this is X and X. This one X came from the mother, one X came from the father. And we know this could be a snapshot of a female. There's no this guy, this could be a male, because in the male would have expected this to be X and one of them to be Y, because the Y came from the father, the X came from the mother. And also when you look at all these chromosomes, it's important for us to just, first of all, just understand to say the chromosomes that are found in an organism, let's say, for example, a cat, a dog, and everything else, we can get a picture, a screenshot of those chromosomes and arrange them according to their height to know that chromosome 1 and chromosome 1 should be paired together, and these chromosomes must carry the same genes. But of course, we know at each of those locus, we can have different alternatives, which we're calling alleles. This is a karyotype. It is just a snapshot of the chromosomes that are found in a species, the complete set of chromosomes. If I had Y here, I would have said this is a karyotype of a male. If there's this, I'll say this is a karyotype of a female. The next question I'm going to ask yourself is a chromosome number important to an organism? Why do we need all these 46? Or maybe, why do I need, can I get a complete set for my father, and then for my mother just get half? Or maybe I love my mother so much, so maybe I should get the 23 for my mom, and then my father should just give me maybe just 10 of them. Is the chromosome number important? Remember, we're saying that we've got 46 chromosomes. 23 of them came from our father. Now that's in reference to a human being, right? 23 came from our father, 23 came from our mother. The question I'm trying to find out here, do we need these individuals to give us the complete set of chromosomes? That is to say, they give us 23 and 23, or maybe one of them can give you 23, then the other guy just gives you 3 or 4, okay, and things like that. So is it important for the chromosome number to be 46? Can it be 45, 47, 48? What happens when that situation occurs? So now we're going to look at genetic disorders. Genetic disorders, these are disorders that come about when there is an alteration in the chromosome number. Apparently, it's important for you and I to understand that the chromosome number is very important. You must have 46, okay? And we understand why that even makes sense. Remember, we're saying that these chromosomes, they're carrying genes. And what are these genes? They're carrying instructions that allow yours and my life to occur, to function, isn't it? So now, if you have got too much of those, it's like an overdose. It's also not good. When you have got less of that, it's also not good. So now, we have a condition that is referred to as monosomy, for example. Monosomy, this is where you have a missing chromosome. That is to say, maybe one parent gave you 23, the other one just gave you 22. So now, instead of you having 46, you have 45. You've got one less chromosome. You can also have more than 46 in a condition referring to as trisomy. In trisomy, it means that one parent gave you 23, the other one was so generous, they gave you 24. And of course, when this happens, either you get more or you have less, you always have a condition that is referred to as genetic disorders. In monosomy, where you have got less the number of chromosomes, an example is Turner's syndrome. Turner's syndrome. Turner's syndrome, most of the time, of course, it affects females because it's due to a lack of an X chromosome. It's a genetic condition that falls under monosomy because you have one less chromosome. Because remember, we're saying monosomy is when you have got less the number of normal chromosomes that you should have. In monosomy, instead of you having 46, you have 45, because in an individual, we expect that you should have 46 XX, but in monosomy, you get 45, you have a missing X. Normally, this happens in females. In trisomy, one of the conditions that is very common is Down syndrome. Down syndrome occurs when you have got an extra chromosome. And remember, sometimes it is just one position. For example, in Down syndrome, it's an extra copy of chromosome 21. So meaning that whatever genes are on chromosome 21, you have an overdose of those genes. Or should I say you've got an overdose of that chromosome. Okay, in an individual, we expect that chromosome 21, there must just be two, one from the mother, one from the father. But then in Down syndrome, you've got an extra copy of chromosome 21, meaning that you've got more than the 46 chromosomes that you need. And because of that, you have this condition referred to as Down syndrome. Of course, it's important for us to understand how are these conditions coming about. It's actually very interesting. I would encourage you to go and read about non-disjunction of these chromosomes, when these chromosomes sometimes fail to separate, during meiosis and things like that, because these are kind of genetic conditions. Now, when you think of monosomy, we can further divide it into two, where you've got partial monosomy and full monosomy. So partial monosomy, it occurs when a segment of a chromosome is missing. Okay, just a segment of a chromosome. For example, the top of a chromosome is removed. The chromosome is there, but part of it has been degraded. Remember, we said the telomeres, those caps that are found at the end of a chromosome, they're there to protect these chromosomes. But sometimes, nature sometimes happened. It could be because of so many reasons. Sometimes even just exposure to carcinogens, UV, and anyway, so many things. You have part of that chromosome being degraded, okay? Or maybe sometimes even just due to age, or maybe even during, DNA repair, something happens to that end of your chromosome, so it gets kind of like chipped out. Now, because the chromosome is there, but you've lost some information, because remember, when you have a chromosome, all that information there is carrying some form of a gene. So if you lose part of the genes on the chromosome, you haven't lost the whole chromosome. Some genes are still there, but because part of that information has been lost, we refer to that as partial monosomy. And the condition that is common is what is referred to as cry du chat, where a child kind of produces this cat-like, sound, because part of the chromosome has been deleted. And the understanding here is that the chromosome is carrying genes. If you lose even a quarter of it, you've lost some information. Okay, you haven't lost, you still have the other combination of the chromosome from your mother, from your father, but because that other alternative or the other gene is not there, you have a condition that is referred to as partial monosomy. An example is the cry du chat, which really normally affects chromosome number 5, the PM. Now, when we say chromosome number 5, what does it mean? I'll take you back to this one. So the chromosome that is affected on when in a condition, genetic disorder, cry du chat, is chromosome 5, when you have deletion of the PM. When you have Down syndrome, it is not any of these chromosomes that are affected. It is an additional chromosome 21. So that is why a karyotype is quite important, because it also allows us to understand these genetic disorders. That is to say, what happens when you have got more copies of chromosome 21? What happens when you have got more of chromosome 5? Even the sex chromosome, what happens when you have got more of the X chromosome? Because there are individuals that have XX and another X, or maybe XX and a Y. What happens when you have got more of those chromosomes? So this is actually a very important picture of our chromosomes, allowing us or helping us also understand some of these genetic disorders. And full monosomy, for example, in Turner syndrome, is a complete loss of the whole chromosome. It is just completely missing, and you've got less of that. So in partial monosomy, you're going to have 46 chromosomes, but of those 46, the chromosome 5, part of it is chipped out. But in full monosomy, the whole chromosome is kind of deleted. It is not even present. So this is the condition that we have in someone who has Down syndrome. So what normally happens in this diagnosis, they're going to create a karyotype of your chromosomes, arrange them from chromosome 1, of course, all the way to your sex chromosomes. Now remember, you can only have either of these, either you are female or you are male. So either in males or female, what happens in a condition of Down syndrome is I've got an extra copy of chromosome 21. Normally you should just have one, not more than one. And normally this happens when one of the chromosomes fails to delink itself, or it's called non-disjoining, fails to separate from the other chromatid. Remember, we have 6 chromatids held by the centromere. At some point in the next topic, you're going to see the centromere has to split, allowing the sister chromatids, to go their separate ways. But when that fails to happen,
when a good thing goes
you can have some of these conditions taking place. Now, this is where I'm going to end. Our next topic is the cell cycle. And I don't want to go into the next topic because we just have one more class tomorrow. And what I wanted to do is to prepare some maybe questions. I'm not too sure if we have any reading material, the tutorial guides, but I can prepare something for you for the topics that we have covered because I know we have a long weekend ahead. Some of you have expressed concerns, worried when we're going to open. I hope we're going to open very soon. And those who are not able to join us, because I've realized our class has reduced. You can imagine we're supposed to 250. So it means even if we're having these classes, most of our colleagues are not, participating for several reasons. So, and I hope when you open, we'll still be able to go back and just, run through this. So I don't want to move too quickly because of that fact. If we're about 100 and 150, I would have said maybe let's continue. But for now, this is a concern that all of us should appreciate. If I complete all the topics, I don't know what's going to happen. It means so many are going to lose out. So tomorrow there'll be no class. We'll continue next week, but I'm going to send some material through Mike to help us. Okay, that one is not for me to answer. I think there's a question. Madam, I'm going to have a quiz. I'm not too sure. That is a prerogative of the coordinator. Yes, the only thing that I can do is to ensure that if I share the material, if you've got a friend who wasn't able to attend class, somehow you're able to get hold of them, maybe they should use the auntie's phone, you can send that material to them on WhatsApp, but I'll prepare that material topic by topic. Otherwise, guys, thank you very much for joining this lecture. I wish you well. And if you have got questions, please reach out. I'm always available. Just reach out. Thank you very much. Have a good day ahead. Bye for now.